Um, so this lecture is it's going to focus on cerebral organoids. This is in reference to the papers we're going to be focusing on as part of the lab section that move into cerebral organoids based stem cell research versus the kind of earlier models of stem cell research the first few papers have been focused on, and I'll describe kind of the difference in this lecture here. So let's just kind of take a look at what we've been kind of looking at so far in the lab section. This idea of taking stem cells from a person, and typically there's two different sources. We have embryonic stem cells, which will come from some kind of fetus, or the kind of new idea to be more therapeutic, have more therapeutic potential, is to take just regular cells from a person that's already developed. It could be a child as well, but a person that is out of the womb, that is alive and still living, and they could be of any age, and simply taking cells, what we call somatic cells. Oftentimes they're fibroblasts, or basically just kind of skin cells, and reprogramming them. That is reprogram. The term is reprogram or de-differentiate them into some kind of to revert them back to a stem cell. So that's referred to as an induced pluripotent stem cell, or an iPSC. Right? And then once you have that, you kind of have this new source of stem cells that came from a person. You can then re-differentiate them into whatever cell type you want into any kind of organ or tissue. So even if they came from the skin, you could potentially make them into some kind of neural stem cell, or into a kidney stem cell, or a cardiac stem cell, or a liver stem cell. And there's all sorts of things you can think about then in terms of not only like replacing tissue, like you can grow new cells and maybe replace the tissue that might be lost from, let's say, liver disease or kidney disease, but also to study diseases, to grow them in a culture, to make models of diseases like liver disease or kidney disease or brain diseases. And then once we can study that more, we can potentially even prevent the disease from happening from the very beginning, or maybe make even better treatments that are easier or less expensive or whatever. Right. So this is kind of what the papers we've been focusing on so far have tried to develop. Taking stem cells, most of them have been iPSCs, some have been embryonic stem cells, and then differentiating them in culture, and then potentially once growing them either as stem cells or as differentiated cells, putting them into an animal, so transplanting them and seeing what happens in terms of curing some kind of disease or alleviating some kind of disease. So this is what we call a 2D model, because what happens is if we differentiate them in the culture, you get this nice examples of these cells that have these nice long processes, combination of dendrites and axons, and they form connections and synapses to each other. And so you can definitely get neurons, and you can definitely get glial cells to differentiate, and you can get interesting networks of cells that, where you actually get true connections, true connectivity between neurons to other neurons, or interactions with neurons to glial cells, right? So this has been, a really good system. This is still a great system to study differentiation or development or interactions of neurons and other neural stem cells or neural derived cells. And so this is definitely still a very feasible, viable model or method to study. But if we're interested in some other questions, like more about actual development of the brain, and trying to model something more about how the brain actually would naturally develop, this is a limited model, right? Because this is not how a brain grows in embryo, in utero. A brain is much more organized. So this is an example of how the cortex develops. It has a very defined set of structures and defined organization, where you have all these layers of cells, and layers form connection, form these scaffolds so that other cells can kind of use them and grow on them and migrate to specific parts of the growing brain. So there's a lot more kind of organization to a true brain that we don't really have in this kind of flat 2D model, where basically we're just growing cells onto a surface, a cell surface, and they're just attached to the cell surface. They're not kind of growing, into each other and developing something more complex. So that limitation, has always been there, and it's been fine. But that's for scientists who are interested in that, trying to learn more about how like a human brain develops. This idea of using stem cells, allows that potential, but the 2D model was limited for that. Right? So what happened? So the idea is, well, if we can kind of, instead of differentiating them into a 2D model, if we can kind of start to mimic that, the true organization, or some kind of organization that is found in the brain. So we have here, instead of just kind of all the different cells mixed together, we start to get some kind of organization where some cells are only found in one part of the culture and other cells are found in a different part, and they continue to divide and grow and get more complex. You can actually develop some kind of, instead of a 2D culture, create a 3D culture, something that looks more like a real developing organ, including the brain. But it's still in culture. It's still just, outside of the body, outside of any animal. It's just simply growing in some kind of incubator. Right? So this idea is actually not that new. This is the advances have come about the last 10 years. So I would say about 2010, 2012 or so is when this new field of organoid research really started to kind of kick off. But it's been around for, since maybe the 80s or even the 90s, this idea, because what people had noticed, what scientists had noticed when in using any kind of cells, and not just stem cells, but when you take cells, that when you co-culture them with a extracellular matrix, so something like laminin or other kind of adhesion molecules that, as we talked about in the lectures, how these extracellular factors can stimulate receptors on growth cones or on stem cells, and the activation of the receptors turns on cell signaling pathways that can promote, a process to become, let's say, an axon versus a dendrite, right? So that the laminin interacts with receptors on growth cones, and only when that interaction occurs does that process become an axon. Otherwise, it's just kind of a random, undifferentiated process. So we know that these outside molecules, these outside proteins, even though they're outside of the cell, they can interact with the cells. And when they do, and if you do it in culture, if you add these extra proteins, these extracellular matrix proteins into culture, they found that actually these kind of very small three-dimensional structures can actually start to appear. So they can get little tubes or tubules or little ducts to appear. So kind of like this, where you get a little bit of this or mini organization, where instead of this kind of just basic flat 2D structure here, you kind of get this little bump in the culture, and that starts to could eventually grow into an even an actual round tube. And so you can use a variety of different extracellular matrix proteins, but there is the most commonly used reagent in research is a compound called Matrigel, which is it's a rich extracellular matrix. So it has lots of different extracellular proteins as part of it, and it's kind of this gel-like substance. And it's a naturally occurring product. It actually is secreted by a specific kind of tumor cell called the Engelbreth-Holm swarm cell line. So that's how it was first discovered. But this is basically now kind of a just widely used reagent for any stem cell work that's trying to get into 2D models, 2D structures. Okay. So this is what we call as kind of a self-organization property of these cells grown in culture. And this is the key difference of what is traditional stem cells, which is what we call as two-dimensional or 2D, just kind of growing onto the surface of a plastic flask or a glass flask itself, versus a 3D culture where the cells are kind of growing with each other and forming three-dimensional structures, and not just growing on to the plastic or glass flask that they're in, or plate that they're in. Okay, so this property I should mention, this kind of just this innate property for cells to grow into kind of very simple three-dimensional structures like tubules and ducts was found. When was it? I forget the exact date, but it was either the 70s or 80s, so decades ago this was known. So people have still were continuing to play around with these properties, and as they started to use Matrigel with stem cells and progenitor cells, they found even more advanced levels of self-organization. So this is this term self-organization, where there's nothing researchers have to do. It's not like they are, that they have to do anything special with these stem cells. All they're adding is this matrigel, this extracellular matrix, and the cells have this intrinsic ability. This is something kind of already pre-programmed into the cell's own development to form these structures when there is this extracellular matrix around. Okay, and when they're stem cells or progenitor cells, they not only then form these structures, but they as stem cells, they're going to differentiate into more specialized cells, and those specialized cells will then kind of recapitulate some of these kind of organs that are found in regular development. So a very I shouldn't say a simple one, but this example here is where this little tube that kind of formed, or little ridge that first formed from endodermal cells, could then further, if you keep growing it, can basically form a mini gut, right? Where you have, this is essentially the inside of what your intestine would be, and so you have these kind of lining of cells here, and you can get even cells that have villi that are kind of moving things around. So you have this lumen of the gut, and you have the different layers of the gut. So you have this kind of inner layer plus outside layers of the gut. So it looks like just a miniature gut, a miniature intestine, right? It's not the full length of what a fully mature adult GI tract would be, or something that complex, but it's a fairly complex structure for simply having grown in culture for a couple of days or a few months without doing much, without having to do much to the cell, simply having it be a stem cell and adding this matrigel to it, and maybe a few common growth factors to help provide some kind of stimulus for differentiation. Okay, so this is what has led to this term organoid. Are these organoids are sometimes called mini organs, where they are these these structures that are miniature organs that are grown in culture. And so they're like an organ, but they're not the full organ, but they're very similar to many features. So let's actually get into what the definition that researchers actually use for an organoid. Okay, so organoid resembles an organ, okay, and implies then a couple of things. So one is that there are multiple organ-specific cell types. So an organoid, by definition, means that you have multiple differentiated cells. So it's not just going to be here. You have an orange cell and a purple cell. So an orange cell represents one type of cell type, and the purple is a different type of cell type. So if we're imagining this to be a cerebral organoid, a brain organoid, we can maybe imagine that the orange are a kind of, maybe they're glutamatergic neurons, and the purple are GABAergic neurons, something like that, right? So there's at least more than just one cell type that's being grown. Okay. Two is that there is some recapitulation of an organ function. Okay, so some at least some function. It doesn't have to be all the functions of an organ, and it doesn't have to be exactly the same way it does that function, but is similar to that. So if we're looking at things like a gut organoid, that there's some level of excretion of enzymes, digestive enzymes. If we're looking at, let's say, kidney cells, that they're able to actually do or kidney organoids, they can potentially even filter blood a little bit. If we're looking at brain organoids, that you can see some kind of neuronal activity, stimulation, or excitation between one neuron to another. If they're muscle organoids, you can see contraction if they're given the right stimulus or calcium or something like that. So they can actually, they have properties of organs in some small capacity. Okay. And then lastly is that they have this specific organization to them that resembles the organization of the true organ. So again, it's not completely going to be, if you have a kidney organoid, it's not going to look exactly like a 2-pound, or however much a kidney weighs, a 1-pound kidney. It's going to, though, have similar layers that a kidney would have, but just maybe fewer cells of those, and just more kind of a condensed, more compact version of a kidney. And it may not actually even look like a kidney all that much, but it may still have the same kind of cell types organized in the same way, in the same kind of layers that might be organized in a kidney, those kind of layers that you would see in kidney cells. Okay. So an organoid formation recapitulates both major processes of self-organization, again the term self-organization, during development. And of that then, there's two major properties of self-organization, which we'll talk about, which are called cell sorting out and spatial restriction. Okay. So this is this idea here where we have differentiating stem cells, and instead of kind of moving, keeping them into a 2D model, they're starting to organize into more three-dimensional structures. And in that three-dimensional structures, they're basically forming what would normally happen in an embryo, and where you get these kind of similar kinds of cell types and similar kind of organization. And as you allow the organoid more time to develop and differentiate, they'll become more differentiated and more complex. The term complex is probably a good way to define this as well, where they essentially then will look, similar to a true organ. Okay. Definition: a collection of organ-specific cells, cell types that develop from stem cells or organ progenitors and self-organizes through cell sorting and spatially restricted lineage commitment in a manner similar to in vivo. Okay, so what are those two terms, or those two concepts of self-organization? The first is cell sorting out. So this is kind of this idea here where you maybe have a mixed heterogeneous population of cell types from your stem cells, and maybe they're just starting to differentiate a little bit, the very first level of differentiation, and in a typical 2D model, they might either stay like this, or maybe they'll kind of form some kind of connectivity to each other, but then a 2D model will kind of stop here, and then in the 2D model there's no real organization to it. They might be connected, but they're not really organizing themselves. Right? Cell sorting out then is this next step where the cells will start to form layers, essentially, where you have one part of the structure will only contain one type of cell, and another part of the structure will only contain a different part of that cell. So this is the cool thing about organoids, is that again, the researchers aren't doing anything. This is simply an intrinsic ability for the stem cells. They will do this naturally. You just give them time, and they will sort themselves out on their own and form these kind of layers on their own. Okay? The cell sorting out is typically due to what we think is, as they're contacting each other, they're using what are called adhesion molecules to bind to each other, or at least to kind of make contact with each other, and then typically to recognize if they have made contact to a different cell. And once they've done that, then they can recognize, okay, are they interacting with their own kind of cell type? So here, purple to purple, or are they interacting with a different cell type, purple to green? And therefore, they can start to make certain types of structures if they recognize that they're interacting with their own, if they're touching their own cell type, or form a different kind of structure if they're touching a different cell type. Okay? Again, totally intrinsic. Nothing the researchers are doing to make this happen. This is just what the stem cells will do on their own. Okay. That then leads to this idea of spatially restricted commitment, is that now once the cells have started to organize, to sort themselves into specific areas where just one cell type exists next to another cell type, then as they start to differentiate, right, this earlier start here is still a pretty early process of the organoid formation. This is still mainly stem cells here, mainly progenitor cells that are just kind of connecting to each other. Early cells. None of these cells here have really committed to a cell fate just yet. But once they start to form that cell sorting out, then as they develop with more time and start to differentiate into more specialized cells, then that where they are in that structure helps to determine what their cell fate will be, right? So the contribution of nearby cells to the cell fate pathway a progenitor will take. Right? So this is kind of analogous to the idea of remember how notch and delta signaling in neural stem cells, where you have two cells touching each other, and both are going to express notch and both express delta, and when there's some kind of interaction, when they bind to each other, the one cell that ends up having more notch becomes the less delta signaling becomes the neuron, and the one that has more delta signaling or more notch signaling becomes the glial cell, right? And so that only happens when they're touching each other. Does that differentiation occur? If they're not touching each other, then you don't get that full differentiation into either a neuron or a glia. But when they're touching, you get that interaction. So this is the same idea here: is that you get a differentiation signal only when a cell is touching or has certain receptors being activated by other proteins that are cell surface bound on other cell types. Okay, so in example here, these cells initially are formed the early stem cells here, and then they're going to differentiate or divide, and they form a kind of a layer on top here, but the cells over here are going to do the same, and so you get a layer of cell types that are organized in such a way that they are on a layer above the green cells, and then these cells will differentiate or divide into a third layer that only occurs after you get this layer forming. Okay, so this is the spatially restricted commitment, so that only these cells will only appear after these cells are only on top of these cells once those appear. Okay, so this is what a real organoid look like. All the images I showed you before were just kind of schematics of an organoid. So this is a true organoid, and on the left here would be organoids from a control patient, and on the right here would be organoids taken from stem cells from a patient. I don't know which disease this is, but you see staining for different kinds of cell types, and you can see you have this ring of one kind of cell type in the middle here, and then surrounding that you get another you get cells that are only staining for this kind of pink stain, and similarly here you get this kind of these ring-like structures that are forming multiple ring structures, but they're all kind of organized in a similar way, right? So this kind of self-organization is a three-dimensional structure, and this is not what you would get just in a regular 2D culture of stem cells. This only occurs once you kind of throw in the extracellular matrix, the matrigel, into the culture dish, and they will then naturally intrinsically form these kinds of structures. And the kind of structures, the kind of organ that will develop depends on the kind of stem cells you're using, or progenitor cells. If you're using straight stem cells, true embryonic, completely omnipotent stem cells that can become any tissue, then you can get a variety of different kinds of organoids, which I'll show you. If you're using more lineage-defined cell types, such as progenitor cell, like neural stem cells versus kidney stem cells, then what the organoids are going to get are going to be more brain-based organoids if you use a neural stem cell, and if you use a kidney stem cell or a kidney progenitor cell, you'll get more kidney-based organoids. So it really depends on what kind of stem cell or progenitor cell you put into the dish, and it will determine what kind of organoids you get. So so like I said, all this is intrinsic to the organoid, to the stem cells. So because of that, then there's not a lot that the researchers have to do at first to make this happen. But because there isn't a lot of additional control that this researchers are adding in, there can be then a wide range of phenotypes that appear. Right, so you can get all sorts of, you get a lot of high degree of variability. High degree of variability then from one organoid to another, because it's spontaneous, it is intrinsic, and anytime you have those kind of spontaneous reactions in a complex situation, just there's going to be chance occurring where one gene or one cell type will kind of push in one direction in one organoid, and one gene or one cell type may take over in another organoid. Okay? So you get a high degree of variability. So there's what I like to call is basically the key driver in this research now is the level of chaos that you're able to control, because if given to itself, you could get a very chaotic, a very just nasty looking organoid or organ structure, and so you don't want to get that, but you still want to have enough complexity, enough kind of intrinsic ability to develop things naturally where it's actually going to mimic what you want to mimic. So it's kind of this idea of trying to get the Goldilocks zone, the Goldilocks spot, where it's not too much, not too little, where you have enough tissue complexity to actually mimic what the true organ would be, but without getting too much chaos where it's just you get way too much variability from one organoid to another, where you really can't then compare results accurately, or you just get a massive, even the organoid itself is just too uncontrolled, and you don't get what you want at all. So this graph here is a really nice one. It shows you, I'll take you through it in more detail on the next few slides. But imagine, we have some stem cells here, and you differentiate them, and these are 4 possible outcomes that you can get from differentiating stem cells, and they range in complexity versus cellular homogeneity. Right, so the more complex a tissue is, that means the more cell types there are, which is what a true organ would be. A true brain would actually have lots of cell types. It has lots of different neurons. It has lots of different glial cells. It has vasculature. It has brain. It has microglia. It has all sorts of cell types are what make up a true organ. And so that's when we say complexity, we're referring to multiples. The more cell types, the more complex. Okay? And so if you have that, then you're going to have very low homogeneity, meaning that you're going to have lots of different cell types. Right? And as you go down this line here, the more you have, just as more get a single cell type, more homogeneous your cell types become, the less complex they become, right? That's the definition essentially. But the more controlled then your experiments are, the more defined, the more predictable your cell types will be when you grow them in culture. Okay, so let's kind of take it from the beginning. So let's start with the simplest form here. The most homogeneous cell type you can get, the least complex cell tissue. Okay? So you can get these are hepatocyte. This is for kidneys. So basically, you get for livers or hepatocyte cells. And so all the cells here look exactly the same. And this is just one coming from one germ layer, which would be endoderm, and you get some kind of patterned organization here, and it's just one cell type, right? And so this is what we call as directed differentiation, where researchers added a few extra specific growth factors or molecules to specifically get this cell type, to just say, okay, we want we're taking these stem cells and we're getting just this. We just want this cell type, right? So it's kind of similar to some of our papers we read so far, where researchers are taking neural stem cells and they want to get just neurons, they want to get just oligodendrocytes, using a very specific mix of media and growth factors and timing to get one specific cell type to arrive in their culture. Okay, so that's this is the least complex but most controlled fate. Okay. If you go one step higher in complexity, we can do what's called, this is a relatively simple organoid. This is from an intestine, where we're only getting one germ layer, of the three. We're taking one germ layer. It's still directed. We're still using, directed differentiation, but instead of just one cell type, we're getting maybe a few, maybe 2 or 3, maybe 4 cell types. So it's not as simple as just, this kind of 2D model. And so you can get a little bit more self-organization, a few more cell types that have kind of organized, but it's still relatively simple. So this is where we can, we've controlled it to a good amount, but there's still a little bit more complexity than would occur just by complete focusing on one cell type. Right? We can take it then the kind of the highest level of where we can see. I shouldn't say the highest, but where we can take it one step level more complex, where we get we're still focusing on one germ layer, but we're letting it be non-directed. So we, as researchers, are basically trying to recapitulate things as, quote, naturally as possible, where we're not directing any differentiation. We're not saying we want just this cell type or just these 2 cell types to appear. We're saying, you tell us, organoid, you determine what cell types you want to become. After we say, okay, we're just going to have just ectoderm. We're just going to pull out the ectoderm stem cells property, but as ectoderm, then you become whatever cell types you want to become. Okay, so it's the level of control has come from the fact that we've only are looking at one germ layer, but then everything else is non-directed. Okay, so because of that, then there's lots of different cell types that come from ectoderm. All the neural stem cells come from that, as well as all your epithelial cells, right? Your epidermal cells. So you get a kind of a mix of this. And so what's interesting is this is actually one of the earliest cerebral organoids that was created. Was a cerebral organoid that actually has essentially growing eyes. These are optic cups here that are part of this kind of mini brain. So even though this is probably one of the most complex organoids that you can develop for a brain organoid, because this is basically undirected, undifferentiated. And so you get lots of different cell types in here. But because it's so complex, it's really hard to study specific things in this organoid. And it looks pretty cool, but it's really hard to actually do anything with this other than just kind of see it develop. Okay? But this is kind of the example of what would be the most complex controlled organoid you get. Most organoids research, as I'll talk about, kind of fit in this range here, where it's kind of relatively simple, just a few cell types so far. That way, because this is where we can still kind of control things and understand things a little bit better, manipulate things a little bit, and less so of this level of complexity of non-direct differentiation. Okay? And then if you were to let things basically go haywire, where it's completely uncontrolled and chaotic, you get what essentially is called a teratoma, which we've read about in some of the papers, but I'm not sure if you've already seen an example of a teratoma. So if you can't tell, this is just a mass of random organ pieces all put together in the same cellular mass. That these all kind of, each part here developed into its own little organ. So you have parts here that look like maybe they came from kidney features. You have teeth that are appearing out of here. You have looks like maybe, I don't know, gut or heart or intestine tissue here. You have, I don't know what this is, some fat tissue. So this is it's just a mess. And this is, the worst kind of thing you would want it to occur, to have happen growing in culture or implanted. This is just, this is would be very dangerous to have implanted or transplanted, and this provides nothing useful in terms of research. So this is spontaneous, non-directed, but you can tell this is the most complex tissue because essentially you have all the tissues here. You have all three germ layers present in this mass here, and therefore it's just kind of it's just a mess. Okay. So that's kind of the basic idea. So let's go into some of the specific details of, if we're looking at cerebral organoids, what are we looking for? What are some things that are going to appear in the papers that we're reading? Okay. So this is the overall idea, kind of a very general schematic here, where we start off with our stem cells in culture and a 2D culture, and then we allow them to kind of naturally aggregate, and you get what's then called an embryoid body. And from there, you can then start to make more complex, specific organoids based on whatever tissue you want, right? So all organoids, no matter what paper we're going to read, all basically start with this embryoid body as the precursor to an organoid. So the embryoid bodies, or EBs, come from stem cells grown in 2D culture, and the embryoid body is the first very early three-dimensional structure you would see from embryoid, but it's still all undifferentiated stem cells. And so this essentially is all still stem cells, but just organized into a 2D structure. So here would be day 0. It's easy for me to tell from looking at microscopy, but this is a two-dimensional structure, or two-dimensional image here. So this would be stem cells grown on a plate. And then after a week or so, they start to form this nice round sphere here. It's a little bit smaller in the middle. So that's an embryoid body. Okay, so this is what all organoids start with: an embryoid body, or an EB, which are undifferentiated stem cells. Okay. Each embryoid body came from a single stem cell. So if this here essentially is a mass of stem cells, what they often do will take this mass here and then triterate it, or basically break it up, or called dissociate into single cells. So we'll see that often, so keep that in mind when you're reading papers. When they say dissociate into single cells, they're taking these clumps. That's how stem cells typically grow in 2D. They grow into these little clumps on the plate, and then they break them up so they're individual. So it's hard to tell, but each little dot here would be a little an individual cell. And so they take those little dots there, break them up so they are completely separated from each other, and then take one of those, put it into a different dish, and have one of those become an embryoid body, which is another mass of cells. But it's in this 3D structure. And so the cool thing then is these embryoid bodies can come from any kind of stem cell, and again, it can be an embryonic stem cell or an induced pluripotent stem cell, so coming from a somatic cell from a patient or a person, and that was dedifferentiated and then and reprogrammed to become a stem cell. Okay. At this point here, once we have an embryoid body, again, this is undifferentiated stem cells, so for the most part, this can become an organoid for any tissue in the body. So brain, heart, liver, kidney, gut. I don't know exactly all the organoids that have been really determined just yet, but essentially any tissue any tissue organoid can be derived from an embryoid body. Okay? If we want to make a cerebral organoid, then we do what was done in vivo during true development, is that there has to be some kind of neural induction, right? To kind of tell these cells here, okay, become neuronal versus something else, versus some other kind of mesoderm or endodermal kind of tissue. So that's what neural induction means. And as we talked about in the early part of the semester, neural induction primarily relies on BMP inhibitors to promote that ectoderm layers to become neuronal, or even to promote some part of ectoderm formation. Okay, so all the protocols we're going to be looking at start with an embryoid body, and then have some kind of neural induction protocol. What's interesting is that this is a very interesting source of debate or experimentation of what's the best type of protocol for induction, and how you can what things you put to induce can get you different kind of phenotypes of cerebral organoids. So this term is very general, and each protocol has their own way. Each group of scientists, each lab has their own specific way that they induce an organoid, or there are specific ways to get specific kinds of organoids based on the induction that they use. Okay. So after the EBs are formed, which takes about 4 to 7 days in culture. So again, here we have our 2D stem cells. Then we we dissociate them, put them into another flask, and have individual stem cells here that then will continue to divide and then form our first sphere-like structure called the embryoid body here. And once we have nice embryoid bodies, then we do a second transfer. We take them and now embed them into this extracellular matrix, the matrigel. Okay, so this blue substance here represents the matrigel that the embryoid bodies are now going to, reside in for the rest of their time in culture. Okay, so when looking at cerebral organoids, after 1 to 7 days after embedding, so after you've embedded it in here, right? Now we can do another count. So once after embedding starts, we can then take another start to say, okay, we can say this is maybe the next day 0, and see how many days after that do things occur. So if we then say 7 days after that, we should start to see what are called neuroepithelial buds appearing from the embryoid body. Okay, so this is typical for cerebral organoids specifically. So this won't necessarily happen in other organoids, but for cerebral organoids, brain organoids, you want to get these little buds appearing. Okay, and you can kind of you want to make sure you look and select and have to do very careful microscopic analysis to pick the right ones, because this is not guaranteed that these buds will form. You can get all sorts of kind of errors for each individual embryoid body. So, for example, this is one where you see embryoid bodies, but you don't see any buds at the same day that you should be seeing, right? Day 14, you should be seeing these nice little buds around, and here you get no buds. So we can't use this embryoid body. Here we get buds, we can see how tiny they are. So they're essentially immature buds, so we couldn't use this. And here we get an embryoid body here, and we're getting these buds, but they're very malformed. We call those cystic buds, where they're buds, but they're not the neuroepithelial buds, right? These are basically cancerous-like buds that are coming off of this. Okay, so you have to be wary, or that you're looking specifically for the good embryoid bodies, and you have to do some kind of quality control to screen out the non-good ones. So it does take a lot of hands-on time under the microscope to pick out the right ones that are growing correctly. Okay. So now, once you have these neuroepithelial buds, you can do either two things. You can either do the unguided differentiation, where you don't try to do anything to the organoid and just kind of let it develop, on its own, and say, here, you tell me what you're going to become. Okay. And so that's where you can get an organoid that looks like this, where this is all sorts of different parts of the brain all coming together, where you get, here's a big ventricle, this is the eye bud, you get cerebral cortical buds here, you get hypothalamic or midbrain areas here. So you get something like that. So that's pretty cool, but it's highly variable, and from one of these to the next, you never know exactly. You're going to get different organoids for every single embryoid body that you've developed. So it's interesting, but more often now, what most research now uses is what's called guided differentiation, where instead of just leaving it to itself, researchers put in a specific combination of growth factors, and also then leave them for a certain amount of time, or then remove them. So it's not just adding a growth factor, it's also removing a growth factor at a specific time that will influence the type of organoid that's formed. So instead of just having a massive, organoid that just has every single brain region, what researchers do now have organoids that are of a specific brain region. Okay, so what's shown here are these are cortical organoids. These just represent organoids that just represent the cortex, the cerebral cortex. You can have midbrain organoids. So these represent organoids, the brain cells that are part of your midbrain, where you have dopaminergic neurons and serotonergic neurons, the VTA, the substantia nigra area here, and then you can have things like the hypothalamus organoid, which just represents that part of just the hypothalamus and nothing else. So these are a couple of examples of organoids that have been developed. Okay, and so this is kind of where most research is moving, or moving towards, or is using our brain region specific organoids versus just kind of a big mass of all the regions together. So you have specifically cortical organoids, midbrain organoids, hypothalamic or cerebellar organoids. Okay. And so this is kind of just a quick reference sheet to kind of show you what I mean by specific growth factors and timing for specific brain regions. So here we have different brain region organoids. We have a cerebellar organoid, midbrain organoid, hippocampal organoid, forebrain or cerebral cortical organoid, and hypothalamic organoid. Right. So we have all these different organoids that are all brain. They're all brain organoids, but they're specific regions of the brain versus just having a big mass where all of these might be showing up together in a big mix or slew like this. So here we have a much more controlled level of differentiation. And so for each brain region specific organoid, we have a unique set of factors. So this one uses insulin and FGF for 21 days, and after that they switch to a different kind of media with different growth factors in them. And this one has different level of timing with different chemicals or reagents added and different media added towards that, right? And so each organoid has its unique growth factors it uses, and typically then there is some kind of then removal or change with time to different growth factors and different media along the way to really kind of push into one specific brain region. So most of the work has focused on forebrain or cortical organoids. That's where kind of the earliest brain specific regions were developed, and so most research in organoids is going to focus on this. The other ones are kind of up and coming, so people are excited about them, but most of the research ends up being kind of forebrain or cerebral cortical organoids. So if we're looking at that in particular, what we can often see then are the appearance of what are called neural rosettes. Okay, so you can see 3 rosettes here, maybe a fourth one, kind of a side view of that one here. But here you can see 3 these rosettes. So these rosettes just mean a nice kind of circular structure that has a couple of circular layers around it. Okay, and what's interesting is that the center of each rosette essentially represents a lateral ventricle. So the center would be just a ventricle. And you can see around each one is a layer of cells that are going to be typically these radial glial cells. So if you remember the lectures on cortical development, you remember that we had here this kind of ventricle here. We had the radial glial cells that surrounded that, and they had these scaffolding processes that extended from one surface to the other, where we had here the layer around the ventricle is referred to as the apical layer, right? And the other side, where they extend to, is called the peel layer. The outer surface is the peel layer, right? And so this is kind of what we're seeing here. So this is a growing embryo in true fashion. And so this essentially is this right here, right? Where this ventricle here is the center here, and you can get this zone of radial glial cells that are around it as the apical or ventricular zone cells here. And you can see the peel zone here is where they're extending to. Okay, so this is a really cool, it really does recapitulate an early portion of a true growing embryo. Okay. So that's just kind of to show an example of one type of brain region specific organoid. But continuing on of what you do once you have something growing like this that looks like it's good. So once we then have those buds develop in the matrigel, we don't keep it in that same culture. We keep it in matrigel, but we move the cells, move the organoids to a more advanced type of flask. So these flasks, all in the earlier days, the first 2 weeks or so, we can grow them in very simple plastic dishes. Essentially, they're just little wells or plates or flasks that you can easily take media in and out of. You put them in an incubator. You let them just sit there, stationary, for those couple of days, and then you can take them out and do media changes pretty easily. Okay? But to keep these organoids growing and living, you have to then take them and put them to a more advanced flask, which is called a spinning bioreactor. And what that is then is, instead of keeping those cells stationary, it's continually moving the media, circulating the media around. So that's why it's called spinning. And so you have a lot more gas exchange into the media. So the spinning nature allows a more rapid exchange of gas and more nutrients to kind of enter into this flask here. Okay? And this is particularly important for organoids because now, as you get these more three-dimensional structures that have more layers and layers and layers of cells around them, that the cells that are more in the center of the structure don't have a lot of have more limited access to the media, to the surrounding media, than the outer layers do, right? And so in a 2D model, all cells have equal access to all the media, but when you start to get these structures, the cells that are more in the middle, they're kind of trapped in the center and don't have direct access to the media anymore. And so you need these bioreactors to help circulate gas and keep the media and nutrients moving more rapidly around the organoids and allow for better diffusion into the center of the media. And if you think about how a normal brain develops, the gas exchange is through vasculature, right? As the brain develops, you get then blood vessels entering into the more center part of the brain and allowing gas exchange to occur through blood vessels. But in these organoids, since we don't have blood vessels come from endoderm? I forget it's meso or endo, but they come from a different germ layer, and so we don't have blood vessels, and so there's no way to get that normal gas exchange that would occur in a true developing organoid. So the spinning bioreactor it kind of is there to mimic or replace what would be there in that would normally occur through vasculature. Okay. And just to kind of give you a sense of that, without that, if you don't have proper gas exchange, you get this necrosis in the middle of the organoid. So here you can see this nice healthy organoid usually is nice and translucent. You can see the light moves through the cells pretty well. But when cells start to die or necrose, they become darker, more condensed, and light does not transmit through them. So you see these dark masses. So you can see these more likely in the center is where the cells are actually dying because they're not getting the proper nutrients. Okay. And so then once you transfer them to a bioreactor, then you can just leave them there for months. And so it does take a lot of media changes, a lot of, you have to keep giving them growth factors and all sorts of different proteins and serum to kind of keep them alive. But now that there's kind of these complex structures, they can survive for months. So here we have these are 50 to 60 days, so they are 2 months old. These are almost 3 months old here. Some articles papers had them 120 days, so maybe 6 months or so. So they can survive for a very long time in as long as they're getting the proper nutrients in the bioreactor. And importantly then, as they're then surviving, they're not just surviving, they're continuing to develop and to differentiate and get more complex. Right, so this would be this early organoid I showed you with the neural rosettes, but here that's so that we can only see one layer of the cortex, the ventricular zone, but then as you get 2 months into it, you can start to see different layers. It's hard to see here, but you can see more layers appearing in the cortex, and then as these cells organoid becomes bigger and bigger, you get more of the cortical layers. Remember, each cortex has 6 layers in it between the ventricular zone and the cortical plate, so you can start to get more and more of those cortical layers in between, not just this kind of very early embryonic looking brain. And I just want to show you the scale of this because as I'm showing this to you, it looks like this may be either super massive. These, how big is how many is a mini brain, or how big is a mini brain? So they're actually still really small. So these are organoids that are about 40 days old. So relatively advanced for what most organoids, cerebral organoid research does. But for comparison, they have this next to a quarter, and you can see each one of these is still pretty small. So they're still microscopic features. They're still microscopic, essentially. You can see them with your naked eye, but they're not. When we say mini brains, they are miniature, like pico nano small kind of brains. Okay. So the last thing I wanted, or second to last thing, essentially is, well, so what's these kind of protocols to develop cortical organoids have been relatively established. There's still a lot of experimentation into refining, specific brain regions and refining protocols so they're not super expensive or take less time, or there's lots of different tweaking going around with these protocols. But what are some of the bigger questions that are remaining? So there's three that most of these review papers kind of talk about. So remember that, so we have these, different brain regions, and they're helpful, they're less confusing than just simply unguided differentiation, where you get this mass organoid of all different kinds of things, optic cups and all, whatever, you don't know exactly what brain regions are in there. So this is better, it's a little easier to understand. But of course, we want a true brain is going to have all of these together, right? So we don't want to just only study these individually. At some point, we would like to see the connectivity. Look at the true connectivity between the cerebellum and the cortex, right? Or the hippocampus and the cortex, or the hypothalamus and the rest of the midbrain. Like that. So we want to see combinations, connectivity of brain regions, but still have it be somewhat more controlled than simply total unguided differentiation. So the first kind of future direction are what we call fusion organoids, where we can take two different brain regions, so one would be a cortical organoid and maybe like a thalamic organoid, for example, and we grow them separately as separate brain region specific organoids, but then we put them together and see what kind of connections they have and see how they kind of talk to each other as many brain regions. So this fusion organoid here representing the kind of full brain cortical to thalamic interaction you would see in a true brain, but represented here. So you kind of grow each brain region separately first, and then put them together and see what kind of connectivity you get. Okay. The next interesting step is to be a little bit more specific in how the organization of the cells. So you may remember that these organoids here are they organize themselves intrinsically based on their own innate genetic interaction, genetic code, genetic blueprint, and so they form whatever structure they want to form. Okay, but a lot of bioengineers or kind of the bioengineering field is trying to take these cells and try to and mimic organs in a more designed, engineered approach. And simply, instead of letting the cells kind of run wild and form whatever organization they want to form, that is like, let's just, we want to make a lung, but we don't want to have this lung mass here. We want to have the same function of a lung using the same cell types, but in a structure that is easier for us to manage if we want to use it therapeutically or to study. So, for example, this would be a true lung, right, where we have these layers of cells here. We have some air, some vacuums here, blood capillaries, alveoli here. So that's how a true organ or organoid would form. Would be this kind of mix of layers here. But the idea of an organ on a chip is saying, well, let's just take that idea and like kind of redesign it into a more what's the right word? Into a more engineered approach, a more rational approach, so to speak. So engineered, where instead of having this kind of random mix here, we're going to take those and we're going to say, okay, this is now our lung, where we have a membrane here, and we have certain cell types on one side, different kind of cell types on the other side, and we're going to have some air flow coming in from one direction, and it's going to come out the other direction. and it's going to function exactly like a lung, and it's going to use the same exact kind of cells that a lung would have, but we have controlled the structure. We have controlled the overall design, the organization of this organ. We're not relying on nature's design for the structure, but the function is the same. Okay, so the function is the same, but the structure is different, is this idea of organ on a chip. Okay, and the last intervention, the last direction, is to then throw in all the other cell types that would normally be found in a brain that are not just brain cells, right? So I mentioned vasculature. Normally, brain, you get arteries and veins coming in, which are critical for brain development and for nutrition and glucose metabolism. And if you're going to model something like stroke, you want to model how brain vessels are in the brain, right? Not just neuronal cells. So vascularization is kind of a new goal to get organoids that have cerebral organoids that have vasculature. Okay? And also throw in cells like microglia, which are not derived from neural stem cells. Remember that they have to migrate into the brain from the yolk sac, and so they're derived from mesoderm. And so if you just have an organoid that's simply derived from ectoderm, theoretically you wouldn't be able to get microglia. But we know microglia are a critical feature of brain health, especially in some brain disease. So we want to have organoids that have microglia in them. So that's one kind of direction of how to make these better models. Okay. And then I'm going to conclude with thinking just like examples of what are we going to do with the organoids, right? So this is really cool in terms of just like being able to do it. So right now, a lot of this has kind of just been like, yeah, let's just see how far we can push this boundary to get these miniature brains develop, to kind of test our scientific curiosity, so to speak. Is there any real potential though? Is there a real use from this beyond just kind of seeing this as really cool? Okay, so there are three major ones that have been listed and that I think are useful, or that are real applications. Okay, so one, like most research that's sponsored by the government, we're trying to model specific brain diseases, right? And so these organoid models have been actually already been proven useful to understanding, given some insights into neurodevelopmental diseases, such as autism and schizophrenia. So making organoids from people that have autism or schizophrenia and comparing their brain development to control cases, and actually seeing some interesting differences, that some of which supported the hypotheses we've had of mechanisms, and which have changed our hypotheses on brain mechanisms for these diseases or disorders. And things like brain cancer, we can grow brain, we can make these and look at it as a real tumor, not just cells in a dish, without having to put it in an animal, right? Not having to actually inject it into an animal and make an animal tumor. We can just grow a tumor in a dish, which is a lot simpler, and we can make more of them more easily. Okay? So these are already been proven useful and are going to be continuing to be proved useful and more developed. And for example, with Zika virus, when that was a big issue a couple of years ago, the earliest studies that showed what Zika was doing to cause microcephaly in infants was using cerebral organoids, and showing how that Zika specifically targets neural stem cells but leaves developed differentiated neurons alone. And that was using cerebral organoids to prove that. Okay. A lot of these review articles say that neurodegeneration can be modeled. So far, that's not been proven, or there's no good model for any neurodegenerative disease, because most of these work on stem cells and development, and neurodegeneration is you have an adult developed brain and then it decays or deteriorates, and so no models have really kind of pushed it towards that end just yet. But that's an idea. So a lot of researchers are trying to get to models like this, models of neurodegeneration or aging. Okay. Another great advance or application is to do high throughput screening. So being able to look for chemicals, pharmaceuticals, genes, again, that are involved in diseases, or that may prove useful in alleviating some diseases. So We can do some of that now, but the better model you have, the more accurate you're likely to get as a result. And we can use these in dishes, and we can test thousands, potentially even millions at a time, versus having to do kind of animal models, which we get maybe dozens or maybe hundreds at a time. We can exponentially increase the number of things we can test in the same amount of time. Okay. And the last one I think is kind of interesting is thinking about evolution, because we can actually see a brain develop in real time essentially without having to do crazy intervention into embryos or uteri, and we can actually look at insights into human evolution. So the idea is to potentially take iPSCs from great apes, right? So just take skin cells from a chimpanzee or from a gorilla and make that an organoid, see how its brain develops, and compare that to a human and see, what are some changes, what are some genetic changes, what are some structural changes, and kind of get more insights into the real differences between humans and other primates. Okay, all right. So that was a overall brief introduction to cerebral organoids. The papers we're going to cover will go into more details into these and expand our horizon into the frontier of cerebral organoid research. But some takeaways from this to keep in mind. Okay, so organoid research, or also called 3D cultures, is a relatively new track, but it's a parallel track to 2D research. So it's not going to replace the traditional two-dimensional uses of stem cells. As we've seen, most of what the papers we've read so far all use 2D, and there's no reason to not use 2D for what their question they're asking. But this is an interesting new track that's tangential or parallel, at least, to the traditional research. So this is not going to replace 2D research, but it is a kind of a new track. Organoids can take on many different levels of complexity, so we can go from very simple to more complex to even more complex, and how complex to make it is really determined on what question you're trying to answer. And so each research question, each study will have different levels of complexity. Just because something is more or less complex doesn't mean that it's more advanced or less advanced. It just means is it able to answer the question that the researchers want to answer. Okay. In terms of the actual protocols or techniques, essentially organoids are very similar. They start similar to regular 2D stem cell cultures. You grow them in the same way as regular ESCs or iPSCs. But the two kind of extra things that are needed are the matrigel, the commonly used extracellular matrix, and then these bioreactors. After a week or 2 weeks, they have to be transferred to these bioreactors to keep all the cells, especially the ones in the center of the organoids, still alive. Okay. And the last point is then, while we have advanced a lot in terms of our knowledge of organoid research in the past decade, 5 to 10 years, there's still many limitations and interesting future directions that this research is taking or can take, both technical in terms of, let's say, fusion organoids or organ on a chip kind of things, but also there's interesting ethical questions which I didn't really raise before. But the idea is, if you keep growing something a more complex brain structure and it gets more and more complex, is there a point where it's going to become conscious, where it becomes, quote, alive, in a way that we don't typically think of just cells in culture? And if that's the case, what kind of research do we think is ethical for that? Or how will we even test to see if, an organoid becomes conscious or not? So there's a lot of, we're at the very, forefront of this technology or this research. A lot of interesting questions being asked. And so, yeah, it's good to kind of keep these questions in mind as we continue to follow this in the future. Okay, that is it. Hopefully that is helpful for reading the last few papers.